Check, 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 check. Hi class, let's focus on our third line of defense, the specific immunity or adaptive immune system. This chapter is a meaty chapter. We're going to break it up and focus on specific components of our third line of defense for our immune system. So without further ado, let's get started. Starting off, let's broadly define what specific immunity is, aka the adaptive immune system. We can only get adaptive immunity after we've been exposed to a pathogen, after we become immune to a pathogen. Our T and B lymphocytes, also known as our T and B cells, are going to be responsible for giving us immunity or activating our adaptive immune system. They're going to go under a series of processes that allow them to react to one specific antigen, or aka immunogen, associated with a specific pathogen. The term immunocompetence refers to our body's ability to react to the countless foreign substances. And actually, there is a number put to it. It's about 100 trillion. We have the ability to produce about 100 trillion different kinds of antibodies. But, you know, in the grand scheme of things, we'll just use the term countless, because 100 trillion is a ginormous number. So we have a lot of variety in the antibodies we can produce. That gives us the ability to have immunocompetence, which means that we can respond to many different kinds of pathogens and have adaptive immunity. An antigen, aka an immunogen, is anything that can stimulate a B or T cell, so our B lymphocyte or a T lymphocyte. Many antigens are going to be a protein or a carbohydrate, aka a polysaccharide, that's going to be on the surface of a bacterial cell or be associated with a virus. If we have anything that's exposed or released, and that is a polysaccharide, it's potentially going to be an antigen. Our foreign molecules that get in our body can stimulate an immune response. And unlike the PAMPs that we talked about in the previous chapter, our antigens are going to be highly individualized and stimulate specific immunity. PAMPs are going to stimulate the second line of defense. So PAMPs can be recognized by those PRRs, those pattern recognition molecules, and be used to initiate phagocytosis of the bacterial cell. But unlike our PAMPs, our antigens don't exist across multiple species. They're going to be unique to a specific kind or specific strain of a specific pathogen. Two features that are going to be associated with our immune response are specificity and memory. Our adaptive immune system is very specific, and our adaptive immune system develops immune memory, or gives us the ability to be immune to a disease. We produce antibodies that can destroy a specific pathogen, but not affect another pathogen. So an example of the specificity is when we make those chickenpox antibodies that don't affect the measles virus. If we take and activate our specific immune system, we'll have some of our lymphocytes programmed to actively respond or recall the initial infection so that we can have a rapid response and very quickly mobilize our immune system to prevent a full-fledged infection and sickness from taking place. During immunologic development, we are going to have the lymphocytes develop and differentiate. Depending on whether we're working with a T or a B lymphocyte, they will develop in different areas of our body. We are going to have presentation of the antigens to our lymphocytes, and then we'll challenge those T and B cells, those lymphocytes, with the antigens. Our challenging of them with the antigens is going to allow us to pre-screen out ones that are going to respond to self-proteins from the ones that do not respond to the self-molecules or self-markers. If that T or B cell responds to a self-marker, so something that belongs in our body, we want to destroy it very quickly. If we don't destroy it, that could be the basis of an autoimmune disease. We only want T and B lymphocytes produced that do not respond to self-markers. Our T lymphocytes are going to mediate cell immunity, and our B lymphocytes are going to be responsible for the production of antibodies. Generally speaking, our T lymphocytes are going to focus on fighting off viral infections. So think of an intracellular pathogen. And then our B lymphocytes are going to focus on bacterial infections or an extracellular infection. All of our lymphocytes, regardless of the type, come from the basic cell type. The same precursor multipotent stem cell produces all of our blood cells. Our B cells are going to mature in a very specialized kind of bone marrow. When you think B cells, I want you to think of bone marrow. And our T cells will be matured in the thymus, so T cell in the thymus. Both of these cell types are then going to move to specific areas of the body to be stored in lymphatic organs. Our B and T cells, while going to be recirculated and moving throughout the body on patrol, migrating between the lymphatic organs and the cardiovascular system, in and out, so that they can be exposed to any potential pathogens that may have entered into our body. This is analogous to a police car rotating on a circuit throughout town, potentially on patrol, trying to mitigate any crime before it could happen. So we can have our precursor lymphocyte, and if it matures in the bone marrow, it will then become a B cell that can be stored in the lymph node or in the spleen. If it matured in the thymus, it becomes a T cell that can then migrate to the lymph node or the spleen. So whether we're looking at a T cell or a B cell, after they mature in the bone marrow and thymus, respectively, they then migrate and rotate between the lymph nodes and the spleen, and will travel through the lymphatic and cardiovascular systems in that process. Our antigen-dependent responses of our lymphocytes can be summarized on this slide, where if we take our B cell and expose it to an activated T cell that's been activated for that specific antigen, the B cell becomes activated. But how does the T cell get activated? We first need to have an antigen-presenting cell phagocytize the antigen and then express the antigen's proteins on its surface. That antigen-presenting cell then carries the bits and pieces of the phagocytized pathogen to a T cell and activates the T cell. That T cell can then activate B cells, and those activated B cells will differentiate to plasma cells and memory cells. The function of an activated B cell that turns into a plasma cell is to make lots of antibodies that go into the bloodstream. Then the memory cells will remain in our body and give us immune memory. Those memory cells, those B memory cells, allow us to rapidly respond to a second or third or fourth exposure to the pathogen. So when we are immune to something, or if we've developed that specific immunity or long-lasting immunity, that is from typically our B memory cells. Our activated dendritic cell, our antigen-presenting cell, activates the T cell, and that activated T cell can then turn into a T helper, a T regulatory, and a T cytotoxic cell. These category of T cells each has specific functions associated with intracellular immunity. Activated T cells can also differentiate into memory T cells, which are going to be able to be rapidly activated and then redifferentiate into our sub-T cell types. And then finally, our antigen-presenting cell can also activate a gamma delta T cell. And activating those gamma delta T cells can also serve to activate additional T cells and trigger the cascade. The gamma delta T cells are the new kid on the block. They have been recently discovered and were added to this current edition of your textbook. They were not present in the prior edition of your textbook. So let's talk about antigen presenting and colonial selection. First, we need to have a pathogen carry the antigen from the first line of defense. So we'll have our resident macrophage migrate to the site of infection. Those macrophages are then going to phagocytize or ingest the pathogen, break it down, initiate the inflammatory response. Then we'll have dendritic cells migrate to the site of infection. Those dendritic cells also phagocytize the pathogen. And dendritic cells, after phagocytizing the pathogen, will decorate the surface of their cell membrane with bits and pieces of the pathogen that they just phagocytized. And it'll carry those bits and pieces of the pathogen to a T and or B cell to activate it. You can think of this as a little boy finding something in the road and carrying it home and showing mom and dad. This dendritic cell will say to the B and T cells, hey, look what I found. And if the T and B cells are pre-programmed to respond to that specific pathogen, they will then be activated. Our gamma delta T cells, unlike just regular T and B cells, can also be activated by the generalist PAMPs, the pattern-associated molecular patterns. So these gamma delta T cells serve as an interface between our specific and non-specific immune system. Those PAMPs, generally speaking, are going to be primarily associated with that second line of defense, but PAMPs can activate the gamma delta cells, T cells, to initiate our third line of defense as well. All the cells in our body have surface markers. These surface markers are used for a wide variety of things. They're used for detection of chemicals, they are used for recognition, so that specific cell types can communicate with each other appropriately, and then finally, of course, they're used for communication as well. Depending on what marker is present, we could have a fully functioning cell or we could have a cell in distress. These markers allow our body cells to communicate and effectively interact with our immune system and allow our immune system to pre-screen what does and does not belong in our body. The genes that code for human cell markers are broad and multivariate. These genes can form the major histocompatibility complexes, the MHCs. These MHCs vary a little bit from human to human to human. And these MHCs allow us to recognize what belongs in our body but doesn't belong in our body. And it can be also associated with some major complications of tissue transplants, because the donor could have different MHC molecules compared to the organ recipient or tissue recipient. Another name for these are also HLAs, human leukocyte antigen systems, but most people, generally speaking, are going to refer to them as MHCs. That's more widely used nomenclature. So if you receive a tissue transplant from someone that has the wrong MHC molecules expressed on their surface, you will reject that tissue or organ transplant. The only time you will have perfect MHC matching or MHCs link with each other perfectly is if you receive a tissue transplant from an identical twin. Generally speaking, if you receive a tissue or organ transplant, particularly the organ transplant, you are going to have to be on immunosuppressants for the rest of your life. There are three broad categories of MHC genes that have been identified. We have class 1, 2, and 3 correspondingly. Hey, that's kind of convenient. Class 1 genes code for the markers that appear on all nucleated cells. So this excludes red blood cells. Red blood cells do not have any nuclei in them. So generally speaking, with the exception of red blood cells, all cells in our body are going to have class 1 MHCs. Class 2 MHCs are going to be on all of our immune regulatory cells, and our B cells and T cells can respond to our class 2 MHCs. And then finally, class 3 MHCs are going to be involved with the complement cascade system. So if we look at a class 1 MHC, this class 1 MHC is a surface marker present on all the cells of our body that have a nucleus. And it's a way of saying, hey, I belong in our body. Other cells in our body that are specifically immune cells can have a class 1 MHC And they can have a class 2 MHC. This class 2 MHC is going to have an antigen binding site, allowing it to respond to specific antigens that are in our bodies or make it into our bodies. Our CD, or cluster differentiation molecules, are a wide category of molecules that stimulate different kinds of immune response. The categories that we're going to focus on are the CD3, CD4, and CD8 cluster of differentiation molecules. The CD3, CD4, and CD8 molecules help to distinguish between different kinds of T lymphocytes and B lymphocytes. They help to differentiate them. We also have lymphocyte receptors. These lymphocyte receptors accept or grasp the antigen in some form. These receptors can also be known as MHC molecules. B cells are going to have receptors that bind to antigens directly on the surface. Our T cells also will have receptors that bind to antigen, but they can only respond to antigens that are presented with an MHC molecule. These receptors have the ability to respond to a wide variety of unique antigens, approximately 100 trillion different antigens. So let's say we have the correct antigen being presented to a B or T cell, so that they match up with the receptor on the surface of that B or T cell. If that occurs, the B or T cell then starts to proliferate, so it starts to clone itself and reproduce very rapidly, and then differentiate. Initially, it goes through clonal selection, so it's going to produce multiple genetically identical cells. Some of those clones are going to be turned into memory cells. Some of those clones are then going to be further differentiated depending on if it's a B or T cell. The responses of our B or T cells differ slightly. They both produce memory cells, but if it's a B or T cell, there could be additional different cell types produced. So let's talk about our T cells. T cells focus on cell-mediated immunity. T cells mature in the thymus, and they typically are going to be the cells that respond to intracellular infections, so viral infections. We'll have our helper, regulatory, and cytotoxic T cells. Helper T cells are kind of like the coordinators or the quarterback of the immune system. They're going to activate macrophages, they activate B lymphocytes, and they also activate cytotoxic T cells. Cytotoxic T cells are going to directly destroy cells of our bodies that have an intracellular infection. So a viral infection or specific bacterial infections that get into our own cells. And those are few and far between. And then finally we have regulatory T cells. These regulatory T cells help to moderate the response of our cytotoxic and helper T cells and help to mediate the immune response. A lack of regulatory T cells has been linked to the production of autoimmune diseases. All of these T cells secrete cytokines. These cytokines are going to help to destroy the pathogens, and it's also worth mentioning that the T cells do not produce antibodies directly. All antibody production is done by the B cell lines. The T cell lines will release those cytokines to destroy the infected cells. So if a human cell is infected with a virus, a cytotoxic T cell will approach the infected cell, and the cytotoxic T cell will release cytokines and cause the infected human cell to be destroyed. If we look at our B lymphocytes, they are going to focus on our humoral or blood-based immunity so that we can attack extracellular pathogens, aka bacteria. So if we activate a B cell, it's going to divide, it'll go through clonal selection, then it will differentiate to memory and plasma cells. A B plasma cell is an antibody factory. That plasma cell is specialized to produce many hundreds of thousands of antibodies per cell that will bind to a specific pathogen. That antibody does not directly destroy the pathogen. The antibody will attach to the antigen that is specific to the pathogen, and it's kind of like a giant red flag or a blinking neon sign or a bullseye. Antibodies don't directly destroy a pathogen, but antibodies make it really easy for our phagocytes to phagocytize the pathogen. I like to use this analogy. You can think of antibodies as sprinkles on a donut. If you have two donuts, you have a plain, unfrosted donut, and you have a donut with frosting and it's covered in sprinkles. Generally speaking, the donut with sprinkles all over it looks a little bit more attractive, and you'll preferentially want to eat that donut. In the same way, if a bacterium is in our body and it's completely covered in antibodies, that bacterium is going to be preferentially phagocytized. It'll be sought out by the phagocytotic cells, so our neutrophils, our dendritic cells, our macrophages, and it'll be destroyed. So, concept check, class. B cells mature in the blank and T cells mature in the blank. Both cell types then migrate to the blank and the blank. So, get me an answer. Pause the video and rewind, check your notes, check your text, or rewind the video. 1, 2, 3, 4, 5. Okay. B cells mature in the bone marrow. T cells mature in the, I should put marrow there. T cells mature in the thymus. And then both will migrate to the spleen or our lymph nodes. So our correct answer is D. That's all we have for this recording. As always, if you have any questions, please post them to the discussion board or shoot me an email. Happy studies.